be performing a transfer as well as a Western blot. As stated in our video before, we did perform an SDS page, and within our SDS page, our goal was to obtain a gel, and the gel was to have protein bands that signified our desired EGFP fusion protein. So what we would do before we begin our transfers, we will go ahead and cut all of these remaining lanes in which we have used to insert our supernatant. After we will go ahead and do that, the purpose of doing so is because we will be placing our gel onto a nitrocellulose membrane. Now this nitrocellulose membrane is known to bind to large amounts of proteins, and the purpose of that is to transfer the proteins from the gel onto the membrane for our Western blot. So we'll see here that this is how our apparatus should be looking like. We do have our Tupperware, in which in our scenario we do have to fill up with sodium bicarb, and this is known as our transfer buffer. So we will fill up our Tupperware in which our cassette is being held with all of our components, which includes our fiber pad, our filter paper, our gel, our membrane, another filter paper, and our following fiber pad. Once we go ahead and do that and place them all nicely together in the order as they should be, we will go ahead and then close our sandwich. And here's our sandwich as well, in which all the components should be placed properly. We will then go ahead and run our gel once more, or run our entire cassette with the membrane as well as the gel inserted. In this case, we will go ahead and run everything into an ice block, the same ice machine that we have used previously for our SDS page. We will go ahead and use that machine, but in this case, we'll run it with ice. We will go ahead and let this run for 1 hour. After doing so, we will then disassemble everything. And the disassemble part, we will go ahead and discard our gel, discard our filter paper, and save our membrane. We will also save our fiber pads. The purpose of saving our membrane is because our membrane has now had all the proteins transferred upon it, so we will be examining this with our Western blot. We will then move on to our Western blotting. Now, our Western blot is where we will begin on adding our blocking buffer and placing it onto a rocker. So here is a rocker which we will use. Now the blocking buffer in our scenario is known as milk, and the reason as to why we'll be using milk is because milk is known to have or carry lots of proteins. The purpose of doing so is that we will go ahead and cover our entire membrane with any proteins, as well as prevent any antibodies from binding non-specifically to any other proteins. Once we have done so, we will go ahead and place it back on the buffer and run with TBST. The purpose of running it with TBST is to go ahead and to remove any antibodies that have not been binded. We will go ahead and then move on to our primary antibody. Our primary antibody is known to specifically bind to our protein of interest, which in our case is the GST and our GST EGFP fusion protein. Our primary antibody will contain a variable component as well as a constant component. Our variable component is known as being our antigen binding site, which will go ahead and bind onto our EGFP fusion protein. Our constant site will go ahead and remain up. We can see here that our primary antibodies are seeking our protein of interest in which they have binded properly to them. After going ahead and doing our primary antibody, we will then rinse. Once again, any proteins that have not been binded will go ahead and be washed off, and any antibodies as well. Everything will be washed off except for those that have been binded onto something. We will then move on to our secondary antibody. Now, our secondary antibody is known to bind to our primary antibody, and it will go ahead and bind to the constant region. Our secondary antibody will also carry an HRP enzyme that when reacted with the substrate, it will create a light substance. We see here that as seen below, we have our proteins of interest with our primary antibodies. Now, if we were to look to it in a gel perspective, we can see our bands, in which we will talk about further. we can see the bands, and we will see our EGFP insert fusion protein, as well as the remaining or the following antibodies binding onto it. After doing so, we will go ahead and then add our substrate, and this substrate will be will bind or react with our HRP enzyme, and it will go ahead and produce us a light reaction. So after doing the substrate and washing one more time, we will then incubate in a dark room for 5 minutes, and finally we will begin our Western blot and seeing our expected lanes. Now with our Western blot, we will go ahead and use a UV light. We see here in our lane 1, we have our ladder. We see here in our lane 2, we have our negative control, which is our PET 41, also known as our empty vector. And lanes 3, 4, and 5 are our PET, GST, EGFP, or the fusion protein all put together. Now the protein samples that contain the GST with the EGFP will reveal a band with a molecular weight in the 59 kb area, which is seen here. We will also see that our negative clones will not express any bands. Now we can also go ahead and use a Western blot ladder in order to help us with this and help us more identify our proteins. Now we will ask ourselves as to why we use Western blotting. Well, Western blotting is used in order to go ahead and measure the different sizes of proteins, as well as being able to distinguish how proteins react to certain diseases or drug treatments that can be